Beef Producer webinar series. Most of you have been with us through the previous 7 webinars, and this is our 8th this evening. Our next to last. I wanted to just probably kind of cut the some of the introduction a little bit short, since I've been through this with all of you so far, and then add a couple of little things as we go through. So tonight, just want to again mention that we've been supported through this entire webinar series, and this will last over into the spring webinar series that we're planning as well, with an emphasis on sustainability through our partnership with the U.S. Roundtable for Sustainable Beef. I've mentioned each time that UT Institute of Agriculture is a member of the U.S. Roundtable for Sustainable Beef, one of the few land grant institutions that are a member, and through our networking opportunities in those meetings that Dr. Bates attends for us, able to partner with Golden State Foods and Nestle to help support us in evaluating the U.S. Roundtable's sustainability metrics through this webinar series. And then again, as we continue to develop our Master Beef Producer training and curriculum and information to deliver to the agents and then deliver to you through one of these methods, whether it be webinar, face-to-face, hands-on, we'll continue to develop how we incorporate the sustainability metrics with the objective of giving you, as a Tennessee beef cattle producer, a competitive advantage in producing a market-ready or a set of feeder calves that are demanded by a market that may shift to a focus on sourcing sustainable calves. And then also, as I've mentioned each time, that part of this is to provide a voice for the cow-calf producer in the discussion around sustainability. The U.S. Roundtable has producers from every sector on that, and that's one of the things that I find very good about the U.S. Roundtable for Sustainable Beef, is that they have input from all sectors, all the way from producer to processor to consumer, food purveyor, in each of those steps along the way. So with that, I'm going to actually switch over to sharing a different screen. I hope this works, because I've kind of given you a little bit of an indication of some of the things that you might see on the U.S. Roundtable for Sustainable Beef's webpage, but I want to call your attention. Hopefully this is showing up correctly, and Courtney, you can let me know if it's not, but the educational modules, the learning modules on the Roundtable's website, and you'll notice that a lot of these are geared toward information that we've addressed already in this webinar series. So Dr. Bates talked about grazing, Dr. Whitlock about herd health, Andrew about transition planning, Kevin Thompson talked about worker safety, cattle handling, those types of things. So we're hitting each one of these and several of the other talks, and certainly the talk that Dr. Beever will give tonight fits into this profitability and efficiencies when we start talking about genetic selection. So again, just really appreciate the opportunity to partner with the U.S. Roundtable for Sustainable Beef, as well as Golden State Foods and Nestle in delivering this webinar series. A couple other things that I'll do quickly, because I think most of you are getting well versed at this, but you'll see some questions here as we get started. Courtney will be helping moderate these questions that'll pop up on your screen. If you're viewing this in the desktop application for Zoom, or on your phone application, your smartphone application, you'll be able to interact with this. And I'll just mention again, if you're viewing this on a web browser, that you won't be able to answer the questions, but Courtney will follow up with a link to a survey where you can answer these questions. This is a sample of what it'll look like. So when you see this pop up on your screen, and then you'll be able to click the most appropriate answer for you. And since we're in a webinar format, since we have so many folks on here, we don't see your video, don't hear your audio, so you don't have to worry about that. But to communicate with us, if you have questions, you can type those in the Q&A box, and I'll be watching that as Dr. Beever goes to his presentation. And certainly there's the chat box that Courtney will be watching and you can put in some questions there. And I think those just go to the panelists and are not broadcast out to everyone in that. So with that, let me introduce Dr. Jon Beever. Dr. Beever's been with us maybe more than a year now. Actually, time's getting by me. But he came to us from Illinois, and he actually grew up as in a beef cattle producing family, has a seed stock background, got his education in molecular genetics, and has spent his career so far looking at genomics and using genomics and genetics to improve the way we select beef cattle, and that geared toward the efficiency and the profitability that we talked about as it pertains to sustainability. So Dr. Beever is the director for the Genomic Center for Advancement in Agriculture here at UTIA, and I've known of his work for a long time, but have been really impressed with the discussions we've had since he's been here, because he can really communicate with anybody on any level, whether it be about just on the ground production, genetic selection, or really kind of high theory, benchtop science, molecular genetics. So it's really neat to be able to talk to somebody about those different things in all those different ways. And I'm excited to hear from him tonight, talking to us about what we need to know about genetics and genetic testing. So with that, Dr. Beever, I'll turn it over to you.
Okay, sounds good. Hopefully everybody can see that. Can you see that, Justin?
Yes, that looks good.
Okay, whoops. Okay, so just a second here, guys. I gotta maybe try to get out of, maybe I can't get out of full screen when all right. So as Dr. Reinhardt said, we're going to talk about, I'm going to give you a little bit of background in genetics, see if I can bring everybody to the same point, point out a few things about genetic traits and how we've come to understand the control of the different traits that we see, and how that's related to variation in the DNA sequence that that animals have in their cells. And so that will lead us into discussion of genomic selection. I think first, Courtney's got some questions for you. So for question one.
Yeah, let me get it pulled up. Okay, you should see question one on your screen now. The question is, when purchasing bulls, is genomic testing an important part of your selection criteria? So just yes or no. And I'll just repeat again, if you're having trouble answering these questions, you will get a follow-up survey tomorrow where you can answer them. And I'll give it a few seconds for everyone to answer. Okay, looks like most of them are in. I'm going to share these results with you.
Okay. So the 33% of you that currently don't, part of the objective, of course, tonight is to hopefully provide the information that maybe causes you to think a little bit harder about doing that, whether or not there's a value proposition there for you. So make sure, and if you have any questions as we go along, to send those to the Q&A box or the chat box so that we can answer them, so that we can maybe get you interested in taking advantage of genomic selection. The next question two.
Okay, you should see question two now. The question is, do you use EPDs to optimize the productivity of your cow herd? So do you use EPDs to optimize the productivity of your cow herd? Just yes or no. Probably about 10 more seconds if you haven't answered.
No jokes tonight, Courtney?
Not yet.
Dr. Beaver, she's been filling this dead space while folks are answering the questions with some jokes in previous nights. So we even had some good ones come
My
in.
jokes have had good reviews, okay? Sharing the results with you.
Okay, so about 57 to 43 yes and no. So we're going to cover that here in the next couple slides. But as Justin indicated, part of the sustainability is optimizing production, and the tools that we're going to talk about tonight certainly provide sufficient information so that you can actually target genetic merit of your cow herd, and you can try to match that to the bulls that you're acquiring or accessing in order to optimize that production and perhaps do it a little bit more sustainably. So with that, this is the foundation part. So I think that if you are in the cattle business or in beef production, hopefully you've thought about what it is that you're attempting to do, what markets that you're attempting to access, or where you're trying to place your cattle once you've produced them. And so I put this slide here at the beginning because genetics can be such an important part of your production system. It's very important to understand what your targets are of that system. You know, selecting animals that are just the most growth or the most milk production or the most marbling or the most ribeye, most is not necessarily optimal. And so I think you really have to spend a lot of time thinking about where your production targets are, what kind of operation you have, and then using the tools in the toolbox to make sure that you do it in an optimal way. And so it's always worth the time to sit down and kind of pencil out where you're headed before you delve into trying to pick the right animals for your production environment and your production targets. So again, to reiterate, some of you guys are in cow-calf production, or many of you are probably in cow-calf production. Of course, that's a totally different market than the guys that are buying calves for backgrounding, or guys that are buying feeder calves for doing calf feds. You know, you might not think about those guys that are feeding cattle or backgrounding and stocking cattle, that breeding objectives are important to them. But what they are taking advantage of where they're getting their cattle from, have to have a certain genetic merit to do what they're intending to do with them at that phase of production. And so again, consideration of what your objective is and how you're going to hit that target market is very important in figuring out how to use these tools that we're going to talk about. So animal breeding is the primary reason to talk about genetics. And what is animal breeding? It's choosing the most appropriate parents to produce the best offspring. And best is highlighted in different color and in quotations there. Again, to reinforce this idea that best is different for each of you, and best has to be suited to your production scheme or your production program. So it's important to talk about how traits are inherited. So I have this the term up here, phenotype. What makes a phenotype? A phenotype is the observable traits or characteristics of the animals that you're working with, whether it be something like coat color or whether it be growth rate, or those are all phenotypes. Those are all traits that we use to inform our decision about what are the best parents to mate together to produce a particular outcome. And I've got a whole list of them here. It really isn't meant to be a multiple choice question, but Only genetics, only environment, mostly genetics, mostly environment, about equal parts of each, or interaction between genotype or genetics and environment. And the answer really is all of the above. We'll talk about a couple traits here next where it is in fact only genetics that controls the trait. But we'll spend most of the time today talking about traits where we have about equal parts genetics and environment, or quite a bit more environmental control than genetics. And I'll try to explain how we estimate what proportion of a trait is controlled by genetics and why that's important here in a little bit. What are we going to use to actually do this selection? So there's another group of things that's not necessarily meant to be multiple choice. Each of you can probably identify some things in here that you use. The last one on there is I give this presentation every once in a while to some young folks that are very heavily involved in the club calf industry, and H is very important for them. I'm not sure why, but that's how it is. Phenotype is, in this case, I'm really wanting you to focus on what that animal looks like from a structural soundness standpoint, traits like that. We don't have a lot of good information for calculating things like EPDs that we'll talk about here in a minute on things like phenotype. There are some EPDs now being generated for feet and leg structure, et cetera. But phenotype is that trait that you want to look at the animal and make sure that everything's kind of in working order. The next few things on the list: performance data, pedigree, breeding values, and genotype or genomics. All of these things actually can be combined, and the end product that we're going to talk about tonight is these genomically enhanced EPDs, or this concept of something called genomic selection. And so again, we take all of this information from B, C, D, and E and combine it into a value of genetic merit for you to use as a tool. I don't know if we'll get to economic indexes, but the economic indexes are based on B, C, D, and E also, and then have the added feature of talking about how they might impact your bottom line from a dollar standpoint. So to start off with, this seems like maybe an odd slide, but I like to use this because what I've got to convince you of tonight is that by understanding how changes in the DNA sequence of an individual change the phenotype of an individual, we can actually make some draw some very good conclusions about the potential genetic merit of that individual. And so this is a really simplistic example. This is one of those traits that's actually 100% controlled by genetics. There's not really any environmental influence here at all. But I like to ask students and in situations like this, producers like you, what's the difference? There's a fairly obvious difference there. One cow's black and one cow's white, right? So I want you to think about it in a little bit different way. This black cow, if I look at a particular DNA sequence within her cells, so cows and humans and pigs and the like, we have something called a genome in each one of our cells. So we have 2 copies of that genome. And that genome is composed of about 3 billion DNA base pairs. And those 3 billion DNA base pairs are made up of are shown by the letters here, A, G, C, and T. So if you were able to look at the DNA of an individual and see it directly, you would see 3 billion of these letters kind of aligned in a linear arrangement down these things we call chromosomes. So if I look at this black cow's DNA at a particular region in the genome, in a gene that's actually called the silver gene, I would see a sequence that would look something like this. If I look at the DNA sequence from the white cow in the same exact region, you can see that for the most part, the DNA sequences are identical, except for right there in the middle, you'll see that there are, there are 3 DNA base pairs missing. And so this huge difference in phenotype, changing a cow from black to white, is controlled by simply the absence of these 3 DNA base pairs at this particular region within the DNA of the animal. Okay, so I think that's pretty amazing. Remember, I think I mentioned it just a couple seconds ago. We have 3 billion DNA base pairs in our genome. You get 1 copy of that from mom and 1 copy of that from dad. So in every cell in our body, we have about 6 billion DNA base pairs. And if I just fool with 3 of them, like I take them out in this case, I can have this huge difference between the phenotype of these animals. One's black and one's white. So I don't have to do a lot. I don't have to change all 3 billion or all 6 billion. I simply have to change 1 or 2 here and there, and I can get quite a huge difference in phenotype. Here's another one. Some of you guys might recognize what's going on in this picture. Again, these are both Charolais bulls. And the question here is what's the difference? Clearly, the bull on the left is kind of a modern day U.S. Charolais bull. The bull on the right, you can see that he's got quite a bit more muscling. And this is kind of an old world French Charolais that actually has the double muscling mutation that we see in other breeds here in the United States. We don't see it very often in Charolais in the United States. The difference between these 2 animals from a muscle standpoint is somewhere between 6 and 8% total muscle mass. So this animal on the right has about 6 to 8% more lean muscle than the animal on the left. And so again, if I turn that around and I say, well, what is that difference really being caused by? I can show you something like this. The bull on the left has a DNA sequence like you would see on top, and the bull on the right has a DNA sequence on the bottom. And the only difference between those 2 animals at this particular DNA sequence is the change from a C to a T. Okay, so by changing a single DNA base pair from a C to a T, I can change muscling or lean muscle production by 6 to 8%. Okay, so this is a very simple example. It's an oversimplification, of course, but the idea here is, again, that I can make very subtle changes in this DNA sequence that can drive very large changes in phenotype. So now we have to talk about the different kinds of traits. The 2 examples I just gave you are actually simple traits, or we refer to them as simple traits. Typically, these are under 100% genetic control, and typically they're controlled by very few genes. And some examples, black, red coat color. Some of you might already be involved in genetic testing for something like black, red, horn polled. Some of you are probably aware of genetic abnormalities within breeds that you use. And these are all traits that are pretty much controlled by these small substitutions in genes and are 100% genetically controlled. On the opposite side, we have traits that we call complex or quantitative. And these are the traits that, unfortunately, most of the things that we get paid for, these are how those traits are controlled. And there's 2 issues here. One, they're controlled by many genes. So unlike our simple traits, things like growth rate, for instance, are controlled by hundreds and hundreds of genes throughout the genome. They're also subject to environmental effects. For instance, nutrition is an environmental effect. So regardless of the genetic merit of an individual, if the nutritional environment that individual is in is not appropriate, they're never going to actually obtain that genetic potential, or they're never going to reach that potential. And so the fact that environment can influence these traits also causes us to have to treat them a little bit differently, and actually winds up causing some difficulties when we talk about selection. And so again, we're going to spend the rest of the time talking about these complex or quantitative traits because these are the traits that you actually get paid for. Pounds of calf at weaning, pounds of milk produced by a cow so that she can have wean heavier calves, growth rate, carcass, ribeye area, and marbling and the like. These are all measured traits that are controlled by many, many, many genes and are the basis of what we actually get money for. So this is just another way to look at these things. Simple traits, typically they're descriptive, red, black, horn pulled. There's discrete categories controlled by very few genes, and there's little environmental influence. Where these quantitative traits are if you can measure a trait, it's probably a quantitative trait. There's continuous variation. So we don't have small, medium, and large weaning weights. We have weaning weights that may, go from 400, 450 pounds to 650, 700 pounds, and everything in between. They typically follow something called a normal distribution or a bell-shaped curve. Again, they're controlled by very many genes, and the environment has the potential to significantly influence all of these phenotypes. So given that, we think about the question, why is it difficult to make genetic progress? And I'd like to give you an example here about these complex traits. So first, remember I said that these complex traits typically have this thing called a continuous distribution or a normal curve or a bell curve. And so let's just say this is a population of animals, and this is the distribution of those animals for, let's say, the trait weaning weight. We treat these traits a little bit different than the simple traits in that we begin to think about them in relative terms. So we begin to think about things in relative terms of things like means. This is the average of the population. We think about things in terms of variance. How much distribution or how much variation is there around this population mean? And we do that because we know that if we want to make genetic progress in a trait, relative to the mean, let's say I want to increase weaning weight. So if I want to increase weaning weight, the animals that I'm going to select for my next generation are going to have to come from the upper end of this distribution. So those animals, the heavier weaning weight, are going to hopefully produce progeny that have heavier weaning weights and drive my weaning weight in the positive direction, so I can produce more pounds of calf at weaning. We call the difference between the mean of the selected parents and the mean of the population something called a selection differential. So this is the amount of progress that we would like to make. So I pick animals that are up here because I'd like my animals to weigh more than the average of the population. Well, let's see what happens. If I take these animals that I selected and I mate them together, they produce a bunch of calves. And these calves also follow this normal or continuous distribution. I've kind of drawn some lines down here that represent the old mean and the selected parents mean. You see that there's been a shift. We've shifted this curve forward, and that was exactly what we intended to do. But in reality, we only shifted the curve forward a little bit. Why is that? Well, the reason why is that even though we selected these animals from up here in this section of the distribution, some of these animals are up here because of environmental influence, and then some of them are up there because of true genetic merit for growth to weaning, let's say. We call this the response. So clearly, we did not get the same response as we were seeking by our selection differential. And by using these two numbers, we can actually calculate something called a realized heritability. And it's not really that important that you know that, but the point here is that by taking the ratio of these two numbers, we can actually get an estimate of how much of the trait that we're selecting on is controlled by genetics. And that's a term called heritability. So again, it's the proportion of the variability in phenotype that is controlled by genetics. And we need to use this because that is what helps us understand how much progress we're going to attempt to make and how much we're actually going to get. So this is a busy slide. I'm going to zoom in on a part of it here. The things that are circled in red are estimates of this thing called heritability. And what I want you to notice here is that on the diagonal in this red box, we have quite a range in this number. We have the high here, a high of 0.49 or 0.48, somewhere like that. And we have some other ones here at 25, 0.25. We even have this one up here at 0.19. What these numbers again are telling us that for yearling height, for instance, this tells me that approximately 49% of the variation I see in yearling height is due to genetics. What that actually also means is that 51% of the variation I see in yearling height is due to environment, okay? So this discrepancy between what I see or what I observe and true genetic merit is the problem that we try to solve by implementing these tools that we're going to talk about. So the idea here is that because there's this kind of this cloudiness of really being able to identify an animal with high genetic merit because of the influence of environment, we have to develop these methods in order to do a little bit better job identifying high merit individuals. Up here at the top of this red diagonal, you notice Caving's direct. Caving's direct is a measure of the difficulty of birth. You see that it's only got a heritability of 19%. So 19% of the variation in Caving's direct is due to genetics. The other 81% is due to environment. And you can probably think of all kinds of things that might influence traits like Caving's direct and even birth weight that are environmental, whether it be cold weather, warm weather, nutrition. A lot of things can influence these traits, and therefore you see these lower numbers of heritability. This whole table is borrowed from the American Angus Association. These are all the traits that are collected information on through the association, and these are all the traits that, of course, have generated EPDs for. So what are EPDs? So EPDs, it stands for expected progeny differences. We'll talk about that when we give you a bull example here in a minute. But what EPDs are is an estimation of an animal's genetic merit based on phenotypes and pedigree information. And it's not only based on an individual phenotype and pedigree, it's based on many, many, many pedigrees and phenotypes. And so what we do is, because selecting individuals all by their lonesome out of a small population, we can make those mistakes in selection. We try to leverage all the data and all the information we have in order to make these estimates of genetic merit. And again, breed associations are the primary entity for the curation of these data. And so the examples I'll be showing you, many of them are from the breed association, in this case, the American Angus Association. So pedigree relationship, why do we care about pedigree relationship? This is a simple 3-generation pedigree of actually a cow that I owned at one time. She was actually as a yearling heifer, this heifer scanned a 10 1/4 IMF. And so we were really pleased with that. And she was phenotypically an outstanding looking individual too. But so I just borrowed this off my breeder website for the American Angus Association. But why do we care about pedigree? Well, we care about pedigree because of the laws of genetics. We know certain things. I know that this heifer got 50% of her genetics from her sire and 50% of her genetics from her dam. Okay, that's it's been true since Mendel come up with the theory way back in the 1860s. I also know that she shares 25% of her DNA with her grandsire, 25% with her paternal granddam, et cetera, et cetera. And I can go back one more generation and indicate that she shares 12.5% of her DNA with her paternal great grandsire, et cetera. Okay. We care about pedigree because if we know that they share a certain amount of DNA and that what is the information contained within that DNA is what's controlling phenotype or what's controlling performance, then by looking at how much DNA these individuals share with one another, and by looking at phenotypic records from all these individuals, we can merge that information together and actually get these estimates of genetic merit. So this actually leverages all the information within the pedigree of an individual instead of just relying on an individual's solo performance by themselves. This is another big graphic, but again, we can zoom in by having the, by sending data into the association as a seed stock breeder, you can actually see the amount of information that's available. Let's just take birth weight there, for example, which is on the second line. The Angus Association has birth weight records for a little over or almost 9 million Angus animals in their registry. They use that information along with the pedigree information to generate estimates of genetic merit on over 11 million animals. Okay, and so this gets at this relationship between data and generating these estimates of genetic merit. I don't have to necessarily collect information on all animals because I can use the pedigree relationship and phenotypic records on other animals to get these estimates. And so you can see that as production goes along, typically we don't get a lot of yearling weights because animals go into they wind up moving herds sometimes and going into the feedlot or something, and so we don't get a lot of yearling data or as much yearling data collected as we might for birth weight and weaning weight. But nonetheless, we can still calculate EPDs based on about half the number of records for all 11 million of these individuals. Dry matter intake, residual average daily gain. You'll notice here that this is a very small number of records. Most people are not equipped or not able to measure this, and so that's why there's very few records. But nonetheless, if you look here, records on about 27,000 animals are used to inform genetic merit estimates in over 1.4 million animals. And so again, this is this idea of taking advantage of these large amounts of data to actually generate genetic merit estimates for individuals. If you were to look at a sale catalog or something like that, and I'll show you that here on the next slide, I think. Again, these are all the EPDs that are generated by the American Angus Association. Calving ease direct, which is CED here, birth weight, weaning weight, yearling weight, residual average daily gain, and so on. I'm not going to go through all of those, but these are basically estimates of genetic merit for an individual, and you can pretty much get this on all individuals that are registered within the association, provided some data has been generated or turned in. This is just the definition of all those traits. Again, there's no reason to wade through all those. You can look at them. I think you probably have access to the slide deck, but just some definitions of what these phenotypes are. So let's look at a set of bulls. I did this on purpose, bull A and bull B. They should look pretty close to the same phenotypically, and that's because it's a cut and paste. So we don't want you to get all confused by saying, well, you know, this one's more structurally sound than the other one. I'm not saying you shouldn't do that. I'm just saying for this purpose, let's consider these two bulls to be identical. But if we look a little bit further, we can see that, of course, by pedigree, they're quite a bit different. And then by EPDs, again, here's all these values that are associated with these estimates of genetic merit. I want you to focus in on a couple things. First, and I will tell you that these two pedigrees and EPD profiles came out of a recent sale catalog. I wanted you to look at the individual performance of these two bulls. Bull A had an actual birth weight of 70 pounds and a 205-day adjusted weaning weight of 737 pounds. And Bull B had an actual birth weight of 60 pounds and a 205-day adjusted weight of 846 pounds. Now, if I were using only individual performance data, I might think that Bull B is, you know, the bull that I need to have those small, light calves that come out with no difficulty, but have these big whoppers that wean off really heavy at weaning, and then of course probably go on to grow out in the feedlot faster and heavier. But if I take into consideration all of the pedigree information and all the phenotypic records collected on all the members of the pedigree, I want you to focus on this. The genetic estimate for birth weight in Bull A is negative 2.8 pounds, where the estimate for Bull B is 2.9 pounds. These things are called expected progeny differences, and what that means is these are relative measures of expected performance, and these two numbers indicate that on average, calves of Bull A will weigh 5.7 pounds less at birth than calves of Bull B. Okay. And so, you know, although individual phenotypic records is another tool for selection, the point here is that EPDs, because they leverage all of the phenotypic and pedigree information that we have available to us, are always going to be a much more accurate indicator of genetic merit or expected performance than individual phenotypic records. Okay, so that's the purpose of this slide here. Now, to tell you the truth, we've done a great job. EPDs have been around. They've not been probably in full implementation, you know, as long as they've been around. But the concept of EPDs was pioneered in the 60s and 70s, the 1960s and 1970s. And I think that if you look at whether it be the scientific papers out there or even some popular press papers out there, the information tells you that we've made a tremendous amount of genetic progress in livestock species and in beef cattle in particular. You know, we raise, I saw a statistic today, I think, we raise the same amount of beef today as we did in 1970 with only with a third less cows. So that's a huge improvement in genetic potential or genetic merit and performance over a 30 or 40 year span. But we still have this issue of accurate selection. And the question is how do we improve that? Can we further increase our accuracy of genotypic prediction? So in other words, can we identify those individuals that have high merit and not have to worry about this cloudy or this gray stuff caused by the environmental influence? And so what this means is if we can accurately identify an animal by direct genotype, then we're going to have a lot higher success in moving our populations forward where we want to. And so the question became about 20 years ago, is there an opportunity for direct selection on genotype? And the answer is yes, which has spawned this field of what we call genomics. And genomics is this idea that if we understand all the things that are going on in the genome of the animals, that we can better understand the biology and better predict genetic merit. And so, again, for the last 20 years, there's been a field of animal genomics which is attempted to understand how these changes in the DNA sequence of different individuals change phenotype. So genomically enhanced EPDs can be accomplished by two ways. So this is determining a genotype of an animal and using that genotype to actually inform the estimation of the EPD by first understanding how DNA variation is associated with changes, or how DNA variation is associated with changes in the traits. So the ability to take variation in traits and somehow link it to changes in the DNA molecule. And so this is a little cartoon. So in this DNA sequence of each of these cows, I can look at different positions and I can see that this cow has an A and this cow has a G and this cow has a T. And then I can use population analysis to try to understand whether or not this change actually influences any of the traits that I'm interested in selecting on. The other way that we can use genomics to enhance EPDs is actually to get an improved estimation of the genetic relationships between individuals. And I'll show you that on a subsequent slide here. This is a pretty average attempt at trying to demonstrate to you that we can indeed understand what genes or what changes in the DNA actually control some of the traits that we're interested in. This is something called a Manhattan plot. Not that you need to know that, but what this graph is attempting to show you, it's attempting to show you where changes in the DNA molecule actually influence variation in a particular trait. In this case, this is a trait called marbling or intermuscular fat. These are data from about 1,600 Simmental steers, where we had complete harvest data on them, including percent marbling. And so what this graph aims to show is that all these dots that you see represent changes in the DNA molecule. So where a A might be turned to a G, or where a C might be turned to a C, or a C might be turned to a T. And these dots are organized across the bottom of the graph here by chromosome. So cows have 30 chromosomes. And so along those chromosomes are all the genes that control these traits. And so over here on the far left, this is chromosome 1, and then next in green would be chromosome 2, and basically they're all kind of aligned in a linear fashion. What I want you to pay attention to is all the dots that are above the reddish colored line here. Wherever these dots exceed this reddish colored line, what the analysis is telling us is that there's a gene or a change in the DNA sequence that is somehow associated with variation in this trait marbling. And I want you to pay attention to these black dots here that are the highest and kind of all aligned here. There's a gene that underlies these changes in the DNA that is actually responsible for about 3.5% of the variation in intermuscular fat deposition. And so by understanding what an animal's DNA sequence looks like for this gene, I can in fact predict about 3.5% of the variation within marbling. And so if I go across here from left to right, you'll notice that there are tens and probably actually even greater than tens, but probably hundreds of genes that appear to have a significant impact on this trait marbling. And so we can actually do that for all the traits that we collect phenotypic information for or performance information for. We can take DNA genotypes and we can marry them together with phenotypic records, and we can identify which of the variations in DNA actually control variation in the traits. And so again, the idea here is that I can, in fact, or we can, in fact, identify how changes in the DNA change the traits that we're interested in. The second part here, this idea of increasing our accuracy of relationships between individuals. On the very first pedigree slide, I showed you that, in fact, an animal inherits 50% of their genetic material from each of their parents. In theory, that's 25% from each grandparent, and in theory, that's 12.5% from each great-grandparent. In actuality, that's kind of not how it works. The parent thing is correct. Each animal receives 50% of their DNA from each parent. But when you get back in the grandparent generation and even the great-grandparent generation, it turns out that because of something called segregation, we can actually, information can actually be not passed on from certain individuals. And what this graphic is supposed to show you is that as we proceed down generations, you notice here, this individual no longer has any of this blue DNA. And this blue DNA is now absent from this individual. And so normally when we would have said that this individual was related 25% to this individual, we can now readjust that estimation to better reflect what the actual relationship is. And we do it by, again, looking at all these markers along a chromosome, all these changes in the DNA. And by looking at those changes in the DNA, we can actually tell what DNA was inherited from which ancestor. And so we can very accurately estimate the relationship between individuals instead of depending on pedigree. And so, for instance, we assume that 25% of the DNA from one grandparent is then present in the offspring, but the reality is that is probably more closer to somewhere between 15 and 37.5, or basically 12.5 and 37.5% of the DNA. And there are some cases, even with grandparents, where there might be zero DNA, as an example here. So by increasing that accuracy of relationship and by being able to use the DNA variation to interpret variation within these traits, we can get these things called genomically enhanced EPDs that increase the accuracy of selection. So how does that translate? That translates in something about understanding an animal's genetic potential better than we could ever before. And this is a graphic. I got this from Dan Mosier when he was at Angus Genetics Incorporated. But the graphic on the left actually shows you the number of cows that have been genotyped in North America. And at this time, there was about 500,000 cows, and you can see where they're located at. So the bigger the circle, the more cows that have been genotyped. But you can see that there's quite a few cows that are genotyped across the entire United States. And of course, many of these cows also have phenotypic records. What this gives us is this added information that's shared over here in the right-hand side of the slide in this table. Let's take our trait birth weight, for instance. If I were to go out and I were to select a bull to bring into my herd, let's say for my heifers, I'm going to want to make sure that he's a calving ease bull so that I don't have to wind up pulling a lot of calves out of my heifers. And so I'd really like that to be quite accurate when I do that. And of course, like you saw in the example of the two bulls, I can select my bull based on his actual performance. But in fact, because that performance is influenced by environment, I might not be making the best decision. And so I want to rely on my EPDs. And now I can rely on my genomically enhanced EPDs. And what this table is trying to tell us is that if I take a DNA sample from a bull and I genotype him for the genetic markers that we currently use, I can generate the same amount of information on that bull as if he were to have sired 23 calves. Okay. So that DNA variation or that DNA sequence information generates sufficient information as if this calf had actually already sired progeny, had already produced calves that were weighed. So think about that. So I can make that estimate the day that bull is born versus waiting 2 years for these 23 calves to be born so that I can measure their birth weight. Okay. So it's actually quite an improvement in estimation of genetic merit. You'll notice here that there's different numbers for each of these traits. And basically this is a reflection of that previous table I showed you on the number of records that are collected for different traits. So birth weight and weaning weight, there were of course quite a few records collected, and we saw that you could generate these estimates of genetic merit on over 11 million animals. Yearling weight, we don't have as many records, but at the same time, we can still make those estimates. Dry matter intake was that trait that we didn't have very many records on at all. And as you get down the table, basically, the lower the number of records that are collected on those traits. You can imagine that carcass traits are a little bit more difficult to measure, not only on the rail, but even by ultrasound. And so that's why most of those are more limited in predictive ability. So I got a summary here. I don't think I've gone way past. In fact, I'm right at an hour. I got a summary here. So selection practice should be matched with production objectives. Hopefully that everybody has already done that, already taken a look at what they want to be doing in their operation, and are appropriately selecting individuals for that operation. I'll tell Justin that could be an entire webinar by itself. I think that it's too easy to get trapped in the more type of phenomena. And I think that if you're going to somebody for consultation on animals to use, you really need to understand what your objectives are, because more is not optimal for most operations, and not profitable for most operations because it's not optimal. So for sure, use tools at hand to select these individuals and make sure that you're doing it consistent with your production objectives. Hopefully, I've demonstrated to you that EPDs are a way to combine a tremendous amount of information to actually get more accurate estimations of genetic merit. Hopefully, I convinced you that EPDs provide more and better information than individual performance records. And it's because, of course, the thousands of records of data that are available, or millions in the case of some of the traits. And then lastly, the reason why we want to go ahead and pursue genomic enhancement of those EPDs is because we can more accurately define the relationship between the animals, and we can leverage the knowledge that we've gained by looking at the associations between the DNA variation and actual performance. And so I hope that you're convinced that using genomically enhanced EPDs or using genomics is one more step in a positive direction and one more tool that can be used for making selection decisions. And then I have question 3 for Courtney.
Sorry, I was muted. You should see question 3 now. The question is, due to this webinar, will you make genomic testing an important part of your bull selection criteria? And then if yes, when will you implement this? I'll give it about 10 more seconds if you haven't answered.
You didn't look up some jokes while Dr. Beaver was talking?
No, I'll tell you, I was trying to be proactive and I already had a joke picked out for every webinar after I started doing that. And after last week's webinar, someone emailed me my joke. So now I can't use it.
Actually, the better answer to that would have been, no, I was so intently listening to Dr. Beaver,
Yeah.
I didn't have time to think of a joke.
That too. All right, I'm sharing results with you.
I'm very happy that 27% of you are already doing it. And I hope I hope somewhere along the way, the 9% can ask a question that helps me convince them that that's the way to go. But these are just one tool in the toolbox you have for selection. So, and I know that it's not realistic that everybody might want to use them. So, question 4.
Oh, sorry, Dr. Beaver. That's probably how I formatted it in your email. There's only 3, so you're good.
Oh, okay. Look at that. No questions. Let's see. Do you guys have a whole thing full of Q&A or?
You know, we've got some really good questions, but I think this is important too if you're going to talk about indices.
Yeah.
Yeah, if that's where you're headed, I'd like for you to go ahead with that. You know, we still have a half hour. So if you'll address this, we've got some really good questions too. I think this will be perfect.
So I didn't know how long if I was gonna go over or not, but I tacked on some couple slides here at the end. And if you remember very back at the beginning, I had something in the list of things that you could use for selection. One was economic indexes. So it's not a new concept necessarily, but it's a growing concept. There are several, there are a handful of economic indexes that are used in most breeds now. And there are even some private indexes that have been developed by different breeders that incorporate different metrics and different economic values. And to get that point across, I'd like to ask this question: is what is the best selection method for genetic or economic gain? And these are some choices: single trait in every generation, single trait in alternate generations. In other words, you select for birth weight one year, and next year maybe for yearling weight, and maybe the next year for ribeye area, and then maybe the next year for something else. Multi-trait selection, so you try to get the best animal you can for lots of different traits, or this thing called index selection. I'm not really going to talk a lot about A and B because it's not really, again, it gets back to this idea of optimums. If you single trait select, then much of the other things just kind of will get all messed up. I think you got to always pay attention to the whole individual, and you can't get focused on an individual trait, or some of those other things will not turn out very well for you. So, for instance, if you're single trait selection on growth rate, and that means you're selected on yearling weight EPD, you're for sure going to run into a birth weight problem, and then a calving ease problem, et cetera. And then the single trait alternate generations, switching back and forth doesn't get you anywhere, because as soon as you make a gain in one area, you're liable to lose it in the next generation. Multi-trait, let me talk to you a little bit more about multi-trait, and this is to express how difficult that is. Oops. I skipped the other slide because I didn't need you to read a paper from the 1960s, but this idea again was pioneered in the 1960s, that the suggestion was that you could make overall economic advancement faster by multi-trait selection. This table, again, it looks kind of busy, but this is actually percentile rank within the breed over on the left. And these are the EPD values that are provided for each individual if they fit into that percentile rank. So I highlight 10% here. So this means that a bull that is in the top 10% of the breed for birth weight, let's say, has an EPD value of negative 1.1 or less. Okay. And for weaning weight, 71 pounds or more, et cetera, et cetera, et cetera. So let's take that more paradigm that I suggested before. And let's say I want to pick a bull that's top 10% calving ease direct, top 10% birth weight, top 10% weaning weight, top 10% yearling weight, and top 10% all the way across the board here. Can't have a show of hands, but I'll let you think about this for a second. If I put in this in my EPD selector that's provided by many breeds, how many animals do you think I would have to choose from? The answer is 0. Okay. So multi-trait selection certainly can move or advance the genetic merit of your herd, but the number of traits that you incorporate into that multi-trait selection very much narrows the opportunity to select individuals, different individuals. So what winds up happening is that you're going to have to do some give and take and put more emphasis on certain traits than others, and really limit the number of traits that you're going to select on in order to better your opportunity to choose amongst many different individuals. But that's where indexes comes in, and indexes allow for that balanced selection of multiple traits. So indexes are based on economic values or dollar values, and there's a weighting that is given, or a weight that is given to each trait in terms of dollar that allows for the calculation of a genetic estimate, allows animals to be quite variable in their EPD profile, yet still optimize economic gain. And so if we just talk about one of these, we talk about maternal weaned calf value. Maternal weaned calf value is a cow-calf operator that might be interested in selecting their own replacement females and selling the majority of their calves at weaning. And so if you think about the traits involved in an operation like that, you got to have a live calf, so calving ease direct is important. You want your cows to calve easy. For calving ease maternal is important. You're selling your calves at weaning, so weaning weight's important, which means milk is important. You want your cows to get pregnant, so heifer pregnancy is important. And as you go down the line here, you want your cows to be functional, so their feet and legs have to be correct. You may not want a huge cow because, of course, she might be prone to eat a little bit more groceries than a cow of a little bit smaller size. So if you take these traits and then you can assign economic value to each of these. How much do I gain if I increase my weaning weight by 5 pounds? How much more does it cost me to feed a cow that produces a pound more milk per day? How much more does it cost me to feed a cow that's 1,350 pounds versus 1,250 pounds, et cetera? How much more often do I have to replace a cow if her feet and leg structure won't allow her to hold up over many years of a breeding program? If you take all those traits and combine them into this single economic value, you can actually rank animals based on that economic value, and there can be huge variability in these individual traits. And so that allows you to get to be able to identify from a larger set of individuals to select from, but still maintain the economic value within your herd. So all these indexes are, of course, based on the same data, phenotypic data and pedigree data and genetic data or genomic data that we've already talked about. It's just they have the added feature of converting that information to an economic value and allowing you to execute multi-trait selection as a combined value, not have to look for those individual EPD values. So the encouragement here is to actually also think about using these economic indexes as a tool for selection rather than selecting on individual EPD values for your bulls. This is the one we talked about, so I think that's it. Yep.
Okay. So you ready for some questions?
Sure.
Good. I think we'll just piggyback off of a little bit about what you just talked about with using the indices. I was hearing a lot about, in general, efficiency in that. And actually, I think as you wrapped up the previous section too, you talked about it's not just producing bigger and putting more inputs in, but the efficiency is really producing more with less. But, as we think about efficiency in general, just wanted to make that kind of clear to everybody here. Had some discussion in the Q&A about it. It's not, when you reference the statistic about producing more beef now with fewer cattle than we ever have, it's not this that we're raising bigger cattle and putting more into them. It's being more efficient. And that's kind of gets to the whole sustainability stuff we talked about, right? There may be something else you want to say about that, or I just want to reiterate that point.
No, I think that's absolutely the case from a sustainability and efficiency standpoint. But I think that I'll go back a little bit, and and perhaps some of the people that answered some of the polls, there's been a kind of a long-standing resistance to the use of some of these tools that have been developed, EPDs, genomically enhanced EPDs, what have you. And some of that resistance has actually not been because people didn't want to try it in the first place. Many people picked it up, went ahead and tried it. And the outcome after trying it, they said, oh, those EPDs don't work. And I'd like to propose that on a fairly grand scale, what was being done was selecting cattle that were inappropriate for programs, that were not optimal, that was not sustainable within that particular operation. So I'll give you an example. Let's just take an example where cows may be running out and it's pretty sparse. I'm not going to want that cow that's got this huge yearling weight EPD if I don't have the groceries to feed her. And so, I use a bull that's got huge growth numbers. I select my replacement females based on their individual performance and their EPDs, and they're going to have huge yearling weight EPDs. And then I feed them, a diet that is insufficient for them to reach that potential. Well, what am I going to get? I'm going to get a bunch of open heifers. And so more isn't better, isn't necessarily better. If the environment's not there to properly let those animals express that genetic merit, then you're going to lose money. And so I think that people said, well, I'm going to select this bull with the best yearling weight EPD. And it caused all kinds of problems because that was not the best bull for their operation. So I think that guys that are resistant still to using EPDs or genetically enhanced EPDs, to go back and think about where that resistance comes from. And I think it's probably in part due to the fact that these tools have to be used to put an animal in its proper working environment.
Yeah, and the same thing can be said about any tool, about a harvester or silo or whatever, right? It just didn't happen to be the thing that fit, and it doesn't mean all silos are not useful, right? Yeah. I'm going to give you a fair warning here. In a minute, I'm going to ask you about genetic abnormalities.
Sure.
But I think before we get to that, talk a little bit about how crossbreeding and heterosis enters into all this. We had some questions and a little bit of a discussion in the Q&A about a crossbreed EPD corrections, the fact that EPDs are specific to comparing animals within breed. How does heterosis affect what you can expect from these predictors, and how does genomics fit into making that more or less accurate?
So to start off with, we go back to EPDs and genomically enhanced EPDs in general. USDA Mark does produce conversion factors every year. They've done that for many years, even before the implementation of genomics. But you can, in fact, adjust EPD values across breeds by using those conversion factors, so that again, that you can make sure that you're matching, the genetics of one breed, let's say the cows, and genetics of another breed, the bulls, to your production program. So that conversion table is available. Most breed associations have it, along with all their other EPD information on the websites. Heterosis is one of those things that still is not well understood molecularly. So in other words, we don't know exactly the mechanism of heterosis on the DNA basis. What we suspected is that each different breed or population, although they may have similar endpoint performance, the genes that regulate that performance are in fact different between the breeds. And so where one breed might have selected for certain DNA variants at a set of genes, another breed has selected for another set of DNA variants at other genes. And even though they might appear similar phenotypically and perform similarly, the basis of that performance is different at the DNA level. And so what heterosis does is it actually brings the variation at those genes together in an animal. And so, the theory is that if you have, positive versions of these genes in two different breeds, and they're different genes, you combine them together, and so that offspring has got more positive forms of those genes. And so there's pretty strong evidence that's what happens. We have learned a lot from our plant colleagues or our crop species colleagues in that regard. Of course, we don't make inbred lines in animals like they can in some plant species, but the basis of some of that is this additive nature of all these different versions of these genes. I think there's some I'll introduce the faculty member. He's not here yet. Some of you across the state will probably soon get to know Dr. Troy Rowan. He's coming to UT in January. And one of Troy's major focuses in his research program is to understand what genes we've actually selected on in different breed populations. And by looking at that, we can actually hopefully gain a better understanding of how much heterosis we might in fact get by making certain matings. The beauty of heterosis, one of the beauties of heterosis is that the traits that have the least amount of heritability, so remember, have least amount of genetic control, actually respond the best to heterosis crossbreeding. And understanding that will be a really key component to really amping up what we can do from a heterosis standpoint.
Yeah, and my personal opinion is, unfortunately, we don't make enough use of heterosis the way it is. But hopefully, if we find the ins and outs of what makes that work so well, then that'll create a snowball to increase that use a little bit quicker. I always compare it to other protein production that are way more efficient, on a calculated basis than we are in beef cattle production. The fact that they make an art out of heterosis, really, and there's very few other protein commodities that are produced without heterosis like we do. So I think there's a lot of room to improve efficiency. Maybe that's a good finding the silver lining in it, right? We have a lot of room to improve. In that answer, you mentioned something about the plant breeders create some line breeding or some inbred lines. Could you address, and I think you have some experience in the Hereford world, so maybe talk about line breeding and how that has been and maybe is effectively used in seed stock production?
So those of you that aren't familiar with the Hereford breed, there's a fairly sizable portion of the Hereford breed is composed of a line of cattle called Line 1. And it's actually the name of the line given to those cattle. But way back in the day when plant breeders, particularly corn breeders, had developed this idea where you could make inbred lines, and then by crossing those inbred lines, you could make these hybrids that would have this accelerated performance due to heterosis. There was an attempt to actually make inbred lines in some cattle, and between Colorado State and the USDA Center in Miles City, Montana, they actually started out with 48 different lines, and every successive generation increased the inbreeding coefficient of those lines. And the reason why there's only Line 1 left today is because all those other lines died out due to the appearance of genetic abnormalities. But what the Line 1 is a success story of is that, in fact, you can by maintaining selection pressure and cautiously watching inbreeding coefficient, you can actually basically increase the homozygosity, which is the similarity of the genes along the chromosome, to an individual fairly highly, and that can act as this resource for getting that heterosis pop when you take it out and put it with a different breed. In the Hereford breed, it actually works quite well even with inbreed. So there are a lot of guys that go and buy bulls directly off the station and take them and put them into programs, and they actually kind of provide a little bit of with inbreed heterosis. But it can work, but the highest inbreeding coefficient that was ever achieved, I think, was a bull. I think eventually he had an inbreeding coefficient of 56%. But that was the highest, I think, that was ever achieved. And of course, you see that it didn't go anywhere just because it's so difficult to do, and you get inbreeding depression and abnormalities, that kind of stuff. So.
Yeah, and that's kind of always been my overall synopsis of it. I worked with when I worked at Mississippi State, there's a producer there in North Mississippi, Hereford producer, that was really good at it. But he was constantly in the numbers and diligently watching all of that. And that's what it takes, I think. Is that a fair synopsis, man? It just really takes a lot of oversight to do that correctly.
Correct. Well, it's a major focus of the entire research program up there at Miles City. That's how much effort it takes.
Right. That's a good point. So in that parlays us into another one thing that I wanted to get to that came up in the Q&A, and then I have one more thing to talk about after that. But the genetic abnormalities, and maybe just start a general kind of a overall take on how you approach managing genetic abnormalities that come up in breeds.
So so of course we've got. I've got a little bit of history. I mean, we started our first genetic abnormality that we identified that what the mutation was, the DNA was in 1997. So I've been doing it a few years. Without a doubt, I mean, there are genetic mutations within the population. Every one of us that are on this call probably have somewhere between 2 and a dozen deleterious mutations floating around in our DNA. And it doesn't cause us problems because many of those things are recessive. So we always have one good copy of DNA doing its job. And the population is large enough and outbred enough that typically we don't have a lot of matings between related individuals, and therefore those related individuals, there's not an opportunity for those mutations to come together. But all populations have mutations that can cause abnormalities. It's how they eventually become observed and what we do about them when they become observed. So populations that are small are more susceptible. So when you talk about, whether or not abnormalities might pop up sooner or later, let's say in, this is probably going to be a off the charts reference, but abnormalities in the Bulingo breed. I don't know if there's any Bulingo breeders out there. I'm sorry if I offend you, but it's a very small population. It's a very new population. It was pioneered in the early 70s. It's a composite. But there's a very limited number of those animals in the United States, and so therefore their genetic base is not very broad. And so the opportunities to observe or to mate individuals together that are related to each other is much, much higher than, say, it would be in the Angus population, et cetera. So typically in smaller populations, we wind up observing those things faster. And that's because of this opportunity to mate individuals together that are more related. In large populations, and again, I'll use the Angus breed as an example, because that population is so large, it takes a long, long time for us to begin to observe those mutations because they have to reach a certain frequency. And what happened several years ago in the case of Angus, and we've done work for Herefords, Shorthorns, and Red Angus and the like, is that eventually the allele frequency or the frequency of that mutation rises in the population to a degree where the opportunity to mate two individuals together that have the mutation is high. And therefore you start getting calves that have these abnormalities. And so that's when you have to implement strategies to try to reduce that allele frequency. You could do it by pedigree selection. Of course, you wind up throwing a lot of animals out because even though they have the same pedigree, it doesn't mean they have a mutation. So that's why we were able to do quite a bit of work on actually identifying which specific mutations cause these diseases. We still every year during calving season, my phone goes off on a daily basis. My email goes off. Get pictures from here, there, and everywhere. Unfortunately, what I have to say most of the time is that congenital abnormalities, which are not necessarily genetic, so abnormalities that occur in animals at birth, are not always genetic. Some of them, in fact, are environmentally induced. And the downside of that is that people often look for an explanation, but there is not one. And the other downside is that for a genetic abnormality, you kind of have to have this wait and see effort. The first calf you can't really do anything about, but when you see the second one and you see the third one, and you see the fourth one, or you see two different breeders from across the country both report one, then you can start figuring out that perhaps this is one that needs to be looked into a little bit further and maybe provide a solution. So I guess my encouragement to producers is that while you're keeping track of during calving season or what have you, always write those things down. I know most of you do, but look for patterns year over year, or talk to your neighbors about things that might be happening in your cow herd if you're using similar genetics. And should something arise that we can provide a solution for, that's what we're here for. But though that's an unfortunate component of any type of system where you're mating animals together to produce progeny, somewhere along the way you're going to wind up with some abnormal ones that are probably due to genetics.
And certainly, something that our seed stock producers across the state have to keep an eye on and figure out how to breed around, or, if those are genetics they want to use, how to avoid having that realized, those genetic abnormalities. But I will say that, again, for our commercial producers, the most effective way to get away from that is to make effective use of heterosis and just use crossbreeding. So it makes it a lot easier, and besides improving all the things we talked about earlier. And in general, just, the comment that there were some questions about, is foot structure genetic, and a lot of questions just, and I get this a lot, is that a genetic thing? And just to reiterate the point that it's genetic by environment, right? I mean, everything is, but some, it'll be more or less on some, and that's the heritability you talked about, right?
Right. So I didn't, I think the current estimates are somewhere around 40%. So about 40% of, so the two traits that the Angus Association is currently collecting is heel depth, or basically heel depth and foot structure or claw structure. And I think both of those traits have about a 40% heritability. So about 40% of the traits controlled by genetics, and that means 60% is controlled by environment.
Mm.
But that's a pretty good heritability to make some really rapid progress in, and I wouldn't doubt that that's one of the traits that can, we can make very rapid progress in with a couple, another 10, 20, 30,000 more records turned in over the next year or two, and the genomic enhancement, that we can solve that problem real quick, I think.
Yeah, I think I've already seen some improvement in it personally, just from looking at the bulls at the bull test and being out and about with producers. Do you happen to know the heritability of temperament offhand?
I think it's somewhere around 20, 25%, something like that.
Okay.
Well, I shouldn't be guessing. I'm not sharing my screen, so I could actually go back and look.
Yeah. Well, I'll just say that we had that question. It seemed like to me that the EPD estimates, at least in Angus, of course, that I've seen for that seem to be pretty true. Again, that's anecdotal, but it appears to me to be.
Okay, so I'm going to admit to being a complete liar. So docility has actually estimated heritability of 44%.
Yeah.
So that's actually moderately high. Right now, the heritability for claw set and foot angle is actually both set at 25%. So, but I still think that we're making a lot of progress in those traits, even with that only 25% heritability. I've seen it. I think so.
Yeah, with so much focus placed on it and discussion about it, I think it kind of been an all-in effort and made some improvement. So last thing, and I gotta say, I appreciate you leaving this much time for Q&A. This has probably been one of the better sessions, webinars we've had for being able to address some Q&A in this way. So the last one I'll ask you something that came up early in the Q&A when you were talking. And again, it kind of relates more to our commercial producers. And we talk about making these changes in things like carcass merit, and we'll just use it as a specific example. But it's really difficult for our commercial producers, the way we market calves, to get an idea of what if we're really making some improvements in carcass merit. And this may be asking you to give some sort of philosophical kind of an answer to this question, but do you have any insight on how we can, as commercial producers specifically, know if we're really making some changes in some of these traits that we don't get to see?
Well, so I think that part of that comes out, part of it comes down to maybe philosophy, or maybe it's a kind of a faith issue, right? Is that there's no doubt that now there's starting to be some literature come out, or some research and scientific literature come out, that is actually showing or demonstrating that we have been achieving more rapid genetic gain in many populations. The dairy industry is a great example. It's of course structured quite a bit different than the beef industry, but we're talking about almost the doubling of genetic gain on a per generation basis by using genomic selection. The pig industry also, of course, it's structured. That those genetics are held in companies where you get some fairly high-powered pyramid production schemes to propagate those genetics, but really rapid advances in some traits that's been really difficult to make progress in over the last 30 or 40 years, particularly on the reproductive side. Unfortunately, because of the diversity in the beef industry, and the records being held at individual associations and things like that, we can't make some of those same kind of statements about the beef industry, but there's no reason to believe that we're not experiencing some of the same exact things. And so I think, those carcass traits are moderately high heritability traits in that 35 to 45% genetic range. And so I think it's just, like I said, it's probably a faith issue, is that we know that the technology is working. We have really good data now. Implementation of this technology is probably a decade ago, 2008, 2010. And we just know that our genetic gain per generation is improving. And so if you're using these tools, you're going to make progress. There's just no doubt about it.
We always, I think, have the tendency to want to know exactly the gain that we're making, or, specific data on our breeding decisions. But sometimes we just don't have that capability, and we can say, we know that we're having more choice or higher grading cattle, more prime. Some of that may be due to instrument grading or whatever, or feeding management in feedlots, but I'd say some of that has to be genetic improvement. Yep. So even if we don't get our individual data back, which we would like to have, and that may be more about what the question was about, there has to be some trust, I guess, in the overall the science behind these. And I think that's a very appropriate statement. So I have 7:06 on my clock. And again, I just want to say thanks, Dr. Beaver. This has been a great session. I appreciate the opportunity to leave some time for Q&A. I think that's been extremely beneficial too, and just some great insight. And, hopefully we can get you on another one of these down the road to maybe talk about some other specific things. So I had saw a good suggestion. I think it was just yesterday. Somebody said we need to close these webinars out with something like Bob Barker used to do with The Price is Right. He'd said, remember to have your pets spayed or neutered. So maybe here it'd be, remember to have your feeder calves cut and vaccinated, and use heterosis in your cows. And EPDs are a good tool. Genomically enhanced EPDs make them better.
Just need to make a whole bunch of t-shirts.
Yeah, that's right. We'll try to boil that all down into one, short saying like Bob Barker did. We're not quite on his level yet, but we'll get there, right? So until then, remember to have your calves cut and vaccinated. All right, so we'll shut her down there. But thanks again, Dr. Beaver. I really appreciate you taking the time to do this. And we'll see everybody else on the next and last one. Everybody take care.